So our last presentation today is, let's see if we can advance here. Oh, there we go. Is Professor Christian Frankenberg, who's a hometown boy here at Caltech. And Christian came originally to JPL, I understand it, in 2010, and he's been here, and then he's been at Caltech the past 7 years. Christian did his PhD in Germany at Heidelberg University. He's a biogeochemist. He uses remote sensing in a lot of his different research activities. So, Christian, please take us home now. Thanks.
Thank you. Can you hear me in the back? Okay, cool. So I have the task to talk a little bit about remote sensing. Only in the end will I really go into soil remote sensing. So bear with me. I'll go a little bit back to the global scale where Dave left us and focus on some parts of the agricultural systems, the overall impact on the global carbon cycle through agriculture, not just through soils, but also photosynthesis itself. That will be my main talk. I want to start with something that crops and basically plants in general have an impact that goes beyond just carbon. It also goes into climate directly through feedbacks with the land, between the land and the atmosphere. And this is from a nice paper from Müller et al. From Peter Heiber's group out of Harvard in Nature Climate Change in 2015, where there was always a big debate of where this warming hole in the Midwest comes from. So these are actually data from 95 percentiles of temperature and precipitation, basically trends therein. This is basically what are the peak temperatures in the summer or peak precipitation. And there was a decline in the Midwest. And there was long-standing debate as to where that is. Could it be related to aerosols or something else? Because there's a cooling trend in extrema in these areas. But this group actually saw that, if this thing works, barely, that it is related to agricultural systems where high photosynthesis and higher transpiration rates through these crops might actually cool the surface. So transpiration that happens during the process of photosynthesis actually acts like a kind of internal AC system, which, if it happens at scale at elevated rates, actually can impact the surface temperature. And this is kind of the opposite of what Piers Seller calls like physiological forcing. So even if CO2 would not be a greenhouse gas per se impacting the radiation balance, it would impact plants in a way that they don't have to transpire as much anymore to get the same amount of CO2 in. Thereby, the land surface would warm just by reduced transpiration rates of plants, basically on a global scale. So this is an impact that goes on top of the radiative forcing of CO2. But this one is the opposite in this case here, where we have enhanced transpiration rates. And all of this goes back to photosynthesis. I took this Google Doodle here that was actually at the birthday of Jan Ingenhaus, who first detected the oxygen generation via photosynthesis, and almost everything that we are talking about is coming from this process. Without photosynthesis globally, we would not have any soil organic carbon that is actually in there. It all comes through the engine of most of the biogeochemical cycles on Earth, which are actually starting through the process of photosynthesis, which A, gives us all the oxygen that we can breathe right now, otherwise we would not have any oxygenic respiration at all, because that only comes from the photosynthetic process. And the CO2 comes in, and carbohydrates come out, but the most important part is basically the water flow through the system as well that is required to basically maintain photosynthesis. To go back to where Dave left us off, I actually like the very simple version of the global carbon cycle that he showed in the beginning quite a lot. So this has a lot more boxes, and without my glasses, I actually start to not see it that well anymore. And the units are always confusing. Like I always, during the talks, I have to recalculate, like, because I like to work in like grams carbon per square meter, because I can kind of relate it to all the other processes that I know from flux towers, for instance, or other things. So this is in petagrams carbon in storage, and then the fluxes are also in petagrams per year. And as was told by Dave, the biggest reservoir of carbon is basically the oceans. So we have like 37,000 petagrams of carbon in the intermediate deep sea. But this is dissolved organic carbon. This is not really reduced organic carbon. The biggest reservoirs on Earth, what we can see here, what sometimes people don't really fully get, is that in vegetation we only have about 500 petagrams stored. And the soils actually store quite a bit more. This is one of the reasons we are here for this workshop. So in soils, we have about 1,500 to 2,400 petagrams. And then in permafrost, we have around 1,700 petagrams of carbon stored. So there's a lot more below ground than there is above ground. Even in tropical systems, it might sometimes be the way. This is sometimes people imagine it slightly differently. And the one process that drives it is basically gross photosynthesis. So this takes up about 120 petagrams of carbon per year. But it's always counterbalanced by respiratory processes that are almost of the same magnitude. So from the perspective of constraining the global carbon cycle, we have the problem that we have these two huge opposing fluxes, which is photosynthetic uptake of CO2 and a huge outgoing flux of respiration that comes through autotrophic respiration of the plants itself, but then also heterotrophic respiration in the soil. And the balance is actually pretty small compared to the total flux. So that's kind of one of the main problems, even though seasonally they kind of are of different magnitudes, of course. And what we mentioned as well is that there's land use, land cover change. So agriculture, on the one hand, we're talking about increasing soil organic carbon in agriculture. On the other hand, they replaced something that has been there before, where typically there was more organic carbon in the soil when agriculture moved in. So this is kind of a source of carbon to the atmosphere, because we assume that agricultural soils are a little bit more poor in carbon. So I think all of the things that we discuss in the future is, or here in this workshop, even if we increase organic carbon in the soil, we always have to make sure that the yields for the agricultural sectors are not being decreased. Because if we have to expand the area of agriculture, then it's kind of things that have to be taken into account as well, because then we would have to convert more land into that. So, and the farmers probably don't like us losing their yield, because that's what they get money from. So this is the global perspective. One thing that I want to mention is, even if you add up all the reserves of reduced carbon in the soil or oil and gas reservoirs and would burn this immediately, it would hardly make a dent in atmospheric oxygen. That actually shows that this curve here is not completely correct, because we have 21% oxygen, so there has to be an even larger storage compartment on geological timescales. In rock formations that is actually not in this image here. Otherwise, we would not be able to get to 21% oxygen in the atmosphere. Because you burn all of this stuff, nothing really will happen to oxygen in the atmosphere. So this is just one part where we have to think about, there are really, really, really million years long timescales for carbon in the Earth system, and that goes into like geological cycles as well. And that's our only way to sequester carbon and a way to actually accumulate oxygen in the atmosphere long enough. And this is from a paper by Kraut et al., kind of a global survey about soil organic carbon in different layers, and on a latitudinal scale here, where we see that above the ground, there's actually quite a bit less carbon stored than below the ground. And that is especially true in higher, like Arctic ecosystems. Even though the productivity is lower, the turnover rates are just slower, so it can accumulate over time the massive amounts, for instance, stored in permafrosts and the like. And microbes are driving these turnover rates. They basically act as a catalyst, otherwise it would take a long time because the activation energy has to be lowered. All of this is a little bit different in agricultural systems because we perturb it all the time. There's tillage, there's other processes, and these are annual plants, as has been mentioned, so it's not a forest that basically is kind of the same tree. The rooting system evolves in time. They just grow, you cut it off, and then the next year will start again. It has one advantage, because we were talking about that we don't really see the soil most of the time. If you have a forest system, how do you measure, even from space, soil organic carbon or anything at the surface? If there's like trees above it, it almost is impossible. At least for agricultural sites, we have the advantage that they are being cut down once in a while, and you can actually see almost bare soil, at least at certain parts of the year. So that is one advantage that we don't have in natural systems, where the bare soil is hardly ever visible. So the motivation why we want to study all this, the plants are doing us a favor, as was mentioned before by Dave. These are the total emissions in time that we emit into the atmosphere. And then land use change is basically this yellowish line here, whereas I think the uncertainties in land use change impacts on the global carbon cycle are still kind of a little bit iffy. For instance, this is kind of, when I did my sabbatical in Heidelberg again, this is kind of a copper stitch from like the 1600s, 1700s. And in the medieval time periods, you saw that lots of areas in Europe and also in other places on Earth were almost completely deforested for various reasons. So it's hard to really go back in time and really see. Actually, now it looks nicer than it was like in 1600. So in some ways, we've gone back to like more vegetation in some of these areas. But to really estimate how land use impacted the global carbon cycle, like in the 16 or 1700, that's pretty tricky. And there might be some uncertainties in there as well. This part is easier. CO2 emissions by industry is pretty easy if you know the carbon content of coal or gas or so forth, that you know how much gasoline you put into your car. It's basically kind of an Excel sheet type of calculation. That should be done pretty easily. But what we see in the atmosphere, as Dave mentioned, we actually see only half of what is being emitted staying in the atmosphere. This is basically the blue part here. A substantial fraction ends up in land with high interannual variations, and the other part is going into the ocean that is in there. The ocean is much more smooth in these estimates here because it's mostly a physical process of CO2 getting into the ocean, because the gradient, basically the concentration gradient changes, and this will not saturate as quickly as basically the land part, and on very long time scales, and also calcium carbonate chemistry and other things will play a role in the ocean, which is kind of causing this to not saturate that early. But understanding the land will be important. What do we know from an observational perspective? The earliest time series basically came from Charles Keeling from the famous Mauna Loa time series, where in the beginning, and now it's kind of head scratching, in the beginning took them some time to actually figure out that the CO2 seasonal cycle in the Northern Hemisphere actually had something to do with photosynthesis in the Northern Hemisphere, where this is basically just the cycle of photosynthesis and respiration going kind of in a different phase, and that causes like the increase and decrease in CO2. If you go into the Southern Hemisphere, this is almost completely flat, just because you don't have enough plants there that have seasonality at the higher latitudes. But how do we know the ocean takes up about almost the same as the land? One method that I actually like a lot is what I call the Keeling combo. So Charles Keeling did a lot of CO2 measurements in the atmosphere, and Ralph Keeling actually pioneered some of the O2 measurements. Changes in O2 are tiny compared to CO2. In absolute terms, they're kind of the same, but in relative terms, they're super, super tiny. And if you look what we should be seeing if we do fossil fuel burning, we take up CO2, so oxygen in the atmosphere should decrease. We should be ending here, but we are actually somewhere here. And then you have two vectors. If you take up CO2 through photosynthesis, you basically change carbon and oxygen. If you change it through ocean uptake, you don't do a dent in O2, just in the CO2. And then you basically have these two vectors, you have to multiply them so that they get you here, and that gives you about a 50-50 term in terms of where the carbon goes to. So this is just one evidence of what the relative fraction of ocean versus land is. So how do we quantify carbon dynamics globally? At the core is photosynthesis, and there's two steps involved, only one of which will really be impacted by CO2 fertilization. The first is we need to harvest light, so we basically absorb photons in the 400 to 700 nanometer range that powers the light reactions, and that's also the place where O2 is being released. The other part is we have to fix carbon, so we use products from the light reactions to reduce CO2, and that will basically take up CO2 in plants. In steps 1 and step 2, they typically have to be coordinated as they work in sequence, at least for regular C3 plants. And if we knew both of them, then the basic photosynthesis model computes the potential rates for step 1 and step 2 and determines the rate limiting step. And all we would have to know is like the partial pressure of CO2 at the chloroplast and the amount of absorbed light. At the global scale, that's what we would have to do. How do we do this for the light reactions? We basically have absorbed light in the photosystem through the antenna system here, and that feeds kind of the light reactions that produce NADPH and ATP. These are the ones that basically then power the dark reactions or the CO2 fixation reactions through two photosystems here. And I'll talk a little bit more about fluorescence later. PS2 actually releases electrons, so a small fraction of the absorbed light is always being re-emitted again. And that is a signal that we can now measure from space and will give you a slightly more direct proxy for the electron transport chain in the light reaction of photosynthesis. If we go to the dark reactions, the Kelvin-Bassam-Benson cycle, basically we use ATP and NADPH to fix carbon. But everything happens just as soil organic carbon decomposition through enzymatic processes. In this case, it's Rubisco. This is this ribulose 1,5-bisphosphate carboxylase oxygenase. And the last part, carboxylase oxygenase, relates to the fact that it can react both with CO2, but also with O2. And that's actually an important point because that is part of the CO2 fertilization story. Typically, people use it in terms of substrate concentrations and Michaelis-Menten kinetics. So this would be the partial pressure of CO2 at the chloroplast level impacts the reaction rates. So this is where if we have higher CO2 in the atmosphere, we will get higher turnover rates. Or plants might decide to actually reduce their Rubisco content and kind of keep the same turnover rates. So there are different trade-offs that plants could do. But the oxygenase that actually through photorespiration always re-releases some CO2 is reduced if we have higher concentrations of CO2. So that's one other part of the CO2 fertilization. And the impact of oxygen is higher the warmer the temperatures are. This is also one reason why CO2 fertilization in the tropics is supposed to be having a little bit more bigger impact than in the higher latitudes. Okay, and last but not least, we also have to move water around. So these limitations of where water comes into the play, plants have to decide how wide to open their stomata to let CO2 in. And this is one part, if we have a higher CO2 gradient between the outside world and the leaf basically in here, plants can decide to close their stomata and still let the same amount of CO2 in. So in principle, in a high CO2 world, plants don't have to transpire as much as they do right now. So they will become more water use efficient. So some of the aspects that we are discussing, that it might be getting drier or the vapor pressure deficit in the atmosphere gets higher, is counterbalanced by the effect that plants actually can reduce stomatal conductance and counterbalance that effect. So how do we quantify the first step of the light reactions? We can measure through remote sensing techniques how much light has been absorbed in the 400 to 700 nanometer spectral range. And those people here who do like spectroscopy of plants, they know that they are pretty dark in the visible spectral range. Even though to us, the human eye, the green still looks pretty bright, but even in the green, they're pretty dark. So reflectances are typically of a canopy less than 10%, even in the green spectral range, which causes this bump where they look green, but they are almost like at 0.02 or 0.03 in the blue spectral range and the red spectral range. And then in the near-infrared range, that what the human eye can't see, plants are actually almost like clouds. They have single scattering albedos of like something like 0.9, so they are highly reflective in the near-infrared range. And this is mostly because chlorophyll stops absorbing around 700 nanometer, and that's the main reason why plants are dark in the visible spectral range, but they are very, very bright, also because of their cell structure in the near-infrared range. And this is just an example where fluorescence is actually being emitted, the signal that we can pick up. So this is, of course, the world as you see. This is familiar. People can look at this and they know what it is. This is how the world looks like in the near-infrared spectral range. You can convert your digital camera. You just have to replace a short pass filter that's on the detector with a long pass filter above 700 nanometer, and you would get something like this. Leaves, this is grass here. They look as bright as clouds. It's very unnatural. So your human eye actually has to get used to it to see what is what. And this is just the direct light here and shaded here in these forest systems here. But if you were to look in the sky, you would actually see that the leaves look almost as bright as the clouds. And this is basically the main basics of the NDVI, or Enhanced Vegetation Index. You just make use of kind of the albedo differential between the near-infrared range and the red spectral range. And that will give you some proxy of vegetation and how much light it absorbs. And this is just one example of kind of the seasonal cycle of this NDVI, which is basically based on the near-infrared and visible spectral range in Africa. And you can also see if plants deteriorate, if they lose some of their chlorophyll, you will see that reflected in NDVI as well. But NDVI doesn't tell us anything about efficiency. It's really just the state of the leaf. How well they can use that light that they absorb, that's highly uncertain. And there's kind of usually a leveling off of the light use efficiency with higher basically irradiation rates and differences between optimal conditions or water stress or some other stress parameters. And that's where fluorescence comes in. That is a little bit more linear to that. It's also not perfect. It's not a perfect GPP proxy, but it is a somewhat better proxy for the electron transport rate in the photosynthetic system. So this is the one part that I'll be talking a little bit about as a remote sensing technique. Chlorophyll fluoresces. That's an intrinsic property of the molecule. So even if you don't have any photosynthesis going on and you just kind of dissolve a leaf in alcohol and kind of have the dissolved chlorophyll solution and you illuminate it with like a blue LED or whatever, you should be seeing a faint red glow. Because the fluorescence emission is overlapping a little bit with the visible, like the human visible spectral range, so it looks like a little bit of a red glow. Would be much brighter at like 740 nanometers. But this is now something that we can observe from space, and I'll spare you the detail as to how we do this. I'll just give you an example now of a growing season simulation over the US. And please, because we talk about agriculture, focus on the agricultural areas, maybe the Mississippi Delta and then the Corn Belt. This is the measurement, and this is kind of the best flux, we call it FluxCom GPP product from the MPI in Jena for global gross primary productivity. I'll start it, hopefully this works. So this is basically February. So we start, the forest systems are starting here, the crops are not active. And now we go into the peak photosynthetic period of the Corn Belt. I'll play it maybe once more. If you just focus on this one, you see that the crops actually have a completely different seasonality compared to the surrounding forests in these areas. Might need to go back and forth. And what you also see is that if you look at Iowa and the Corn Belt and look at peak fluorescence values across the US, it actually happens across the globe. Every time I look at like these peak productivity values, at least in terms of fluorescence, they are almost always crops. So that happens. And they have a different seasonality. Typically, the growing season is much shorter than for forest systems. And that actually can even cause a substantial impact on atmospheric CO2, because you have a more spiked growing season that can actually create a deeper seasonal cycle in the atmospheric CO2 in the northern hemisphere. And this is just one example first on the global scale. So you see it a little bit with the color scale here, a little bit clearer here. But you also see some of the deforested areas in South America at the edge of Brazil here, and some of the crop areas in Argentina that are bright, like lighting up basically during their growing season. So the impact of crops on global productivity is getting stronger and stronger, I would say. They take up more land, and they're actually some of the most productive ecosystems. And when you look at like flux tower sites in the middle of Iowa, even now you probably get like daytime CO2 values somewhere around 340, 350 ppm. Kind of almost pre-industrial, and then early in the morning it's like 800 or 900. So you have a huge diurnal cycle of CO2 in crop areas because they take up CO2 so quickly. This is an example where we can actually use fluorescence data to estimate the crop yield. USDA is great because they provide like county level statistics in units that I don't like, but that's another thing. That's kind of history, like bushels. So you have to compute bushels for different crops into like grams of carbon, whatever, so that there's prone to error in many, many different ways. But we can see if we have the USDA crop productivity maps in grams per square meter here and the Tropomi kind of summertime values for fluorescence, they actually correlate really, really nicely. The only discrepancies that we here see is the ratio of C3 crops per county. The main reason is that C4 crops have a higher efficiency, so even if the electron transport rate is the same, C4 crops would do more photosynthesis. And that's one reason why we see this discrepancy here. If we level this out and take like into a linear regression the fraction of C4 crops or C3 crops in there, we basically get a quasi-linear relationship here. One thing that I should mention, you see this net primary productivity in grams carbon per square meter per year. It can go up to like 800 grams of carbon per square meter per year productivity for plants. For comparison, within the atmosphere, if you were to integrate all the carbon molecules in a square meter to the top of atmosphere, it's about 1.6 kilograms of carbon per square meter in the atmosphere. These numbers are not too dissimilar, so they actually take up very, very fast. If it would be like an isolated column, it would take out half of the CO2 of that atmospheric box. And then we go into other parts. We looked at the same thing. Ian did this, who's probably also here. In the future, we will have more drought, but we will also have more flooding. And for 2012, a lot of the people who study crops probably have seen the dramatic drop in crop yields in 2012 in the drought. This is an example for 2019 where it was actually too wet. So it can sometimes be too wet and sometimes be too dry. In 2019 was too wet that actually caused the delay in the planting, or it was already planted and was basically flooded, so it didn't really grow very well. And we see that time period between 2018 and 2019 with dramatical differences here, where we basically had a really big reduction in fluorescence in 2019 compared to 2018. And that is directly related to the crop area ratio, so it's mostly related to basically the maintained agricultural system. So these will be the problems in the future for crops, droughts, and more flooding, more extreme events on either side. Sometimes too much, sometimes too little water. And this is just an example of what the crop productivity in the areas can actually have an impact on CO2 as well. This is kind of a fancy animation where we have fluorescence underlain here, and then you see CO2 vertical profiles here near the surface, where you see the dramatic uptake of CO2 here in the crop areas in the peak season with like lower values below 390 ppm during the time. And we typically see this behavior in the CO2 seasonal cycle and the fluorescence seasonal cycle. So when dCO2, basically if fluorescence is highest, the dCO2 dt is maximum, kind of the steepest decline here. This is typically what we see. What we also see is trends in peak productivity across the globe, some of which are related to crops. And we also see a deepening of the seasonal cycle in atmospheric CO2. And we can try to relate this to different systems where we can model the seasonal cycle amplitude in atmospheric CO2, and it has increased if we use some of the metrics that we have before and simulate this through the atmosphere. We see a seasonal cycle amplitude increase in some of the areas that are related to crops in here that actually impact now the global carbon cycle as well and might even propagate into some of the higher latitudes. So crops are playing an increasingly important role so that we will also need to understand them to understand the global carbon balance. So crops are playing this important role. Where does the carbon go? They take up a lot, but how much is really staying in the soil? That's the big question. And how can we quantify this? So this is a paper now from Kai-Yu Guan, where we'll steal some slides from. What I mainly talked about before is now GPP, so the photosynthetic uptake of CO2. It's going into biomass, and then it goes into crop yield and harvest. That's one big difference to natural systems. Typically in a forest, everything that's taken up is respired in place. For crops, like Iowa, like this area of Iowa is a huge carbon sink. But then they export all the crops across the world and it's being respired elsewhere. That actually has to be taken into account if you do like carbon balancing across the globe. Some of this stuff might actually end up as food waste and go into landfills. And we have studies on that actually matters because it might be permanently sequestered that way as well. So it's not always, it depends on where we put the food waste. There's ways of actually mitigating things. The big question now is though, how much do we lose through respiration? And especially, how does an increase here translate into an increase in the soil organic carbon here? And what are the other management practices in agriculture that can actually perturb this part here so that we can get more stored in this aspect here? And these are just a few slides on what we know about soils. This was kind of one of the earliest studies that I found on like the properties of soils and organic matter in soils for reflectance spectrum in the visible and near infrared. It's a paper from 1997 by Ben Dor et al. From Israel during a controlled decomposition process. So they have a t equals 0 time period here, and then after 3, that's kind of a year of decomposition process of that specific soil. We see change in the reflectance spectra, gets darker overall, but then some of these features are also different. What I like about this paper, they actually associate some of these spectral ranges with specific pigments, with lignin or with the kind of the CH stretch of aliphatic versus aromatic compounds. So they don't just do like machine learning and regress some of these features against like the organic carbon compounds, but they actually specify in which spectral range do we actually see true properties of organic matter of carbon near the surface. And they associated with all of those. So this was one of the earliest study. And there's lignin. We talked a lot about lignin. It's pretty recalcitrant for CO2 decomposition. And these are the things that we could, in principle, measure near the surface. But as Nimrod said, how deep do we actually probe? What do we see? We see a little bit of roughness where we sometimes have like exposed soil mixed with like residues from the crops before. So it's always kind of a little bit of a mixed component. What we see, and Caillou's group here worked a lot on different aspects across the globe using soil samples, and they grouped basically some of the reflectance properties by soil organic carbon content, lower than 20, which is more like mineral soil, and higher than 120, organic soil. And you see features in the reflectance spectrum in there. At the same time, we have these aspects of kind of management practices that can change organic carbon. Tillage is mostly like the top 20 centimeters. In no-till systems, you basically leave everything on there and you don't till it in. You might have cover crops or no cover crops, and just visually you already see that there's differences in the color of the soil depending on the organic carbon compound. The big question is, how do we relate kind of the surface reflectance properties of soils, agricultural soils, to what's below ground? Is it reflective of like the top 20 centimeters if it gets tilled around? What happens to like below 20 centimeters and the like? Because this is something we will never ever really see from space. The question is how it's similar. The similar problem to SMAP, soil moisture active passive. We basically see 2 centimeters soil moisture. How does that relate to root zone soil moisture? It's a tricky question as well. But there's hope that we can actually see kind of the fraction of basically crop residue cover, and this could be done here in this case here, again from his group, airborne derived residue cover versus ground residue cover. And there's some spectral ranges that fit this process very well. This is a machine learning technique to basically regress it against. So there's some hope that we can actually see what's on the ground near the surface. How that relates to the subsurface is a different question, also because some of these aspects are kind of compounded by other impacts, because soil moisture changes the brightness of soil. Green vegetation might still be on top of it. That changes the reflectance spectrum, crop residue as well. And that is all different from like particulate organic matter near the surface. So the challenge is we are blind to that stuff in principle. So remote sensing is blind to the subsurface. How do we relate what we can observe to total carbon storage? And that's the trickiest part, and the challenge for our workshop will be kind of along those lines. Steve has tackled this one here. The spatial variation within any given field can be larger than year-to-year changes in soil organic carbon. That's kind of a statistical problem, where even if we were to measure the heck out of it, it's going to be tricky, because you measure here or there, the natural variance in space is so high that it is hard to measure trends without actually maybe going 5 years, every 5 years, or every 10 years to detect trends. So soil sampling is infeasible in a short-term quantification method, but more well-positioned to the soil organic carbon baseline. That also makes me think about models. Some models might be bad in getting the absolute carbon right, but are actually better in getting the trend or the change right. So we have to maybe separate and discuss like model structure, model parameter errors, and not always say like just because they don't get the total carbon right, but they have processes in there that actually can at least give a fractional response to change in the future right. We should keep this in mind. Remote sensing techniques, particularly hyperspectral, have shown potential to monitor soil organic carbon, but as was said before, we really only detect soil carbon at the surface, not the soil profile to full depth. For water, if you think about kind of SMAP, we have GRACE, which is great. Gives us like the water column integrated to the deep earth. For soil organic carbon, we don't have anything like this. Would be nice, but we don't. Crop residues, green vegetation cover, and soil moisture can have a confounding impact on spectral signals, so there would need to be some techniques to actually fix this. And with that, I'm done. I was actually wearing a Keck KISS workshop t-shirt here when we did like some flux tower studies in Iowa, which was fun. And sometimes you see like 2-year-old residues, because this is a no-till system, so you see the corn from like 2 years ago still on the same rotation. And that shows that sometimes carbon takes a long time to decompose. Thank you.
Chris,